So today we are going to discuss about the one more and very common categories of chemical compounds which are present in plants as such, and their derivatives are also found in the plants. And then because of the one moiety that is present is derived from sugars or carbohydrates, or we can simply call it glucose. So that is derived from that portions, and different types of sugars are associated. That's why we call this category of compound as glycosides. Okay, that contains actually two moieties. One is carbohydrate moieties that is sweeter in taste compared to one other moiety, and that moiety can be different in different kinds of or different categories of glycosides actually. The second moiety, apart from the sugar moiety, we call it non-sugar moiety. Okay, and we also called it aglycone. The sugar moiety, we call it sugar moiety or glycone as such. The another name is glycone, and the another name for non-sugar moiety is aglycone. And this aglycone moiety can be can be varied or can be different in different categories of glycosides. It can be steroid, it can be saponins moieties, it can be uh, anything apart from carbohydrate. We call it aglycone moiety. So these both moieties are connected together, are attached together, and that makes a various therapeutic categories of compound, and which are present in many species of the plants, and that we call glycosides as such. So now if you just see the definitions and introductions of glycosides are non-reducing organic compounds that on hydrolysis, means when breakdown with water, that actually gives two moieties. One is glycone, another is aglycone, and along with breakdown of with water, we can also increase this hydrolysis portion by addition of acids, alkalis, or enzyme associated hydrolysis part. Okay, and so the breakdown of sugar and non-sugar moiety, that depends what chemicals, what reagent you are adding along with water as such. Okay, to increase rate of reaction, we add acids, we add alkali, and we many times also use various enzymes as such. And then these categories of compound, they get dissociated into two parts. One is sugar part, that is actually made up of one or more than one unit of sugars, and second is non-sugar part, that is actually made up of anything apart from carbohydrate, and that we call aglycone, or also called as genin, GENIN. Okay, which is mentioned over here also, right? So next we see that these categories of compound, or these are actually condensed compound. When two sugar and non-sugar portions, they come together and then they bind. So these condensed compounds are called as glycosides as such, and they are again the secondary metabolite. They are not the primary metabolite, they are again the secondary metabolite as alkaloids are as such. Okay, because they are not required by the plant in their normal or in their daily physiological processes. So these compounds are required by the plants in some special cases only, or definitely they are very significant in medicinal properties and medicinal activities in various pharmacological actions which they show on animals as well as on the human, as well as in the human beings. That's why we are studying about these categories of compound which are present in the plants as such, right? Then if I just tell you what are different classes of chemical compounds or glycosides are present in the plants, then if you just see this, then we have, so if we discuss various classes of glycosides and various divisions of glycosides and various categories of glycosides, then we see firstly we can divide them on the basis of the condensation, on the basis of in between, atom that is present. Okay, if sugar and asugar portions, they both are connected together by the different atoms, and that atomic linkage actually we call it glycosidic linkage. This kind of linkage actually we call it glycosidic linkage. And this glycosidic linkage, it can be like through the oxygen, it can be through carbon, it can be through sulfur or nitrogen. Mainly these four categories of atoms are present between the two bulky molecules. One is sugar and another is aglycone molecule. Okay, and actually the aglycone molecule is much bulkier, or if I talk about their molecular weight, they are heavier compared to the sugar portions. Sugar are usually one sugar, two sugars, or three sugars they are present. Okay, over here the sugar means they are not like you have studied about monosaccharide, disaccharide, polysaccharide. So they are not polysaccharide sugars. They are maximum, they are trisaccharide sugar. Maximum times we got or we studied about trisaccharide sugar as such. Okay, and then if I just tell you why they are so significant, why what is the property which is making them which is making glycosides as one of the favorite drug molecule to study, or we have already lot of drugs into market from this category, because we have two moieties as such. One is sugar. Okay, and you know sugar is very water soluble. Sugar is very much water soluble. It can dissolve, it can absorb through our cells, through the solvents. The maximum solvent which is present in our body is water only, so it can pass through that. Okay, it can properly distribute through that, it can properly dissolve through that, it can properly absorb through that. But the second portion, aglycone, and then you see the connection between glycone and aglycone, and they are like linked together by these atoms or by glycosidic linkage. So sugar, it can dissolve itself. Aglycone portion, it can produce the various therapeutic actions as such. That's why this combination, this friendship becomes one of the most significant feature in pharmaceutical sectors to study. And we have already a lot of medications into the market like decoctions and many others which having glycosidic linkage, which having this categories of compounds. Okay, so aglycone, which is responsible for various therapeutic actions. Then the problem of bioavailability, the problem of dissolutions, distributions, the problem of metabolism, and and and, and like the availability of drug at the site of action is resolved by the association of sugar that is present. So wherever sugar goes, it takes the aglycone molecule with it. And then because of enzymatic action, because of change in the pH, that can be acidic, that can be alkaline, then these two portions get get free from themselves, or they get separated. Now the aglycone portion, it can produce the therapeutic effects, and glycone portion, it helps that aglycone portion to reach up to the site of the action. That's why they have very good drug candidates. Okay, and that's why like we try to synthesize these kind of chemical compound in the laboratory, which is having both. If you just try to see this, look at over here. If you just see, then you will find out, let me start the screens slide, so then you can also see with the help of animations in this, that over here, if you just see this ring structure, okay, this is actually the glycosidic linkage. And now there is one glucose as such. Now there is one glucose as such. And now we have one more category of the compound which is higher in molecular weight. We call it aglycone. And this totally is the structure of human's favorite flavor, which we call vanilla. Okay, we use it in various like chocolates, ice cream, vanilla flavor like butterscotch, and so many things. So this is actually a glycosidic compound which is again present or originally derived from the plants only. Okay, this is present in green vanilla pods, and definitely if you just taste the glucovanillin, this is bitter because of the aglycone portion. Okay, and then that the order are there, but identically if you just use it in very less concentration, then you can see the vanilla flavor, or uh, like in chocolates, and definitely you also have like appreciate that flavor which is there. And then nowadays we are totally synthesizing this compound. Okay, because of its flavoring property, we are totally have gone into the mass production of this compound in our laboratory. And this very simple molecule is actually helps us to tell you that where is glycosidic linkage, where is a glycone portion, where is glycone portion. Okay, and that clear it. And now if I just ask or raise this very simple questions from the first point that is mentioned, this is which kind of glycoside? This is again O glycoside. This is O category of glycoside, because oxygen is helping these two portions to bind together. Then similarly, if carbon is present at oxygen, then we call it C glycoside, we call it S glycoside, we call it N glycoside, and definitely we will study the each and every category with their examples as such. Now, if only one sugar is present, if you can see over here, then we call it monosaccharide. Okay, the another example of these are salicin, which is present in salix bark also, and only one sugar portion is present. If two sugar portions are present, okay, then we have example of diosmin. If three sugars are present, this is most significant compound which we are using in various heart problems, and CHF, congestive heart failure, in ischemic heart failure, in when heart is not able to give the proper cardiac output, that in that cases we are using deoxin. Okay, digital is deoxin. So that actually having three sugar molecules as such. And then apart from this, we have we can divide the glycoside on the basis of its nature, means presence in nature, whether primary glycoside, secondary glycoside. Primary glycoside is actually present in plants as such. Now, as soon as you introduce some enzymatic actions or some hydrolysis, what happens? The sugar from 3 sugar, it start dissociating, it start becoming 2 sugar, then it become finally at 1 sugar. And then if you just continue the hydrolysis portion, that 1 sugar also it get dissociated, that glycosidic linkage it break down, and then 1 sugar also it get released, it get free from the aglycone portion. So on the basis of the nature of the glycoside, we can call primary glycoside, we can call secondary glycosides. Primary glycosides are originally present in the plants as such, okay, like purpurea glycosides. Purpurea means digitalis purpurea, that's from the plants as such. Okay, and then secondary glycosides that actually resulted by the actions of hydrolysis. And then we have like category of decoction under this under secondary glycosides as such. Okay, and that's why like we use green vanilla pods, we use glucovanillin vanillin as such, because of the order and because of the characteristic smell, because of the characteristic taste also. They suppress the bitter components which are present in medicines, and that's why we add it in, and it in protein X powder, where like people do not want to take because of the ammonical smells or nitrogenous smells which is present in protein. Sorry, some network interruption was in between. So the next type of division of glycosides are on the basis of the linkage which is present, and that's can be alpha glycosides, that can be beta glycosides. It depends on the stereochemical structures also. Okay, the association of sugar with a glycone portion. Definitely, likewise, we divide the alkaloids on the basis of the family, on the basis of the species, on the basis of source from where we are obtaining them. Similarly, we can divide glycosides as such. The glycosides can be digitalis glycosides, it can be senna glycosides on the basis of the source or the plant source from where we are getting it, from where we are obtaining it. Okay, then we can divide glycosides on the basis of... Okay, voice is not coming. we are discussing that the glycosides can be alpha and beta on the basis of their linkage. It can be divide again on the basis of the botanical source, definitely again on the basis of their pharmacological actions, therapeutic effect. We can divide analgesics glycosides, purgative glycosides, cardiac glycosides, and then we can have bitter glycosides. We can have glycoside which you can use as a diuretics as such. Okay, glycosides, licorice glycosides, the glycosides which can have the which can you can use it in various like throat actions, and then various inflammatory actions and infections. That can be one more category. Then we can divide glycosides on the basis of the chemical nature of aglycone. Aglycone can be flavonoid, it can be steroidal, it can be aldehydyl, it can be saponins. So likewise we can divide. Just if you see the next part, that is characteristics of the glycosides. If you just see them and then how they look like. Okay, they can be amorphous or they can be crystalline, but maximum time they are solid as such. Like maximum time alkaloids are also solid. Similarly, glycosides are also solid. They can be amorphous, they can be crystalline. They are non-volatile in nature. Okay, very similar characteristics as alkaloids are. They are bitter in taste. Usually they are bitter in taste because unless until the sugar is free. If we just remember our knowledge from carbohydrate topic, polysaccharides are not sweeter in taste. They are not sweet in taste compared to oligosaccharides, compared to disaccharides, compared to monosaccharides. Polysaccharides are blunt in taste, or there is no taste as such, because unless until the sugar portion is free, and then our receptors which are present on tongue, they can feel the sweet taste. Similarly, because over here the sugar portion is binded, that's why they are usually tasted as bitter. But as you think, as the name suggests, glycosides, okay, if at very faster rate, if they can release from the aglycone portion, they can be sweeter in taste. There is one simple question. If you just chew the portion of bread without any curry or something, it's will not give the sweeter taste in the initial part, but slowly as you start chewing it will also appreciate, or you can also appreciate the sweeter taste into that, because the polysaccharide portion is now getting divided into the smaller oligosaccharide portion, disaccharide and monosaccharide portion. The amylase which is present in our mouth, that helps in dissociating the carbohydrate portion. Similarly, at first instant they are usually bitter in taste, definitely because of the presence of what? So your sugar portion, they are soluble in water, and they are soluble in polar organic solvents also. They are soluble in water and polar organic solvents, and this solubility portion makes them very favorable for therapeutic actions, because aglycoside portion that is not soluble in water, glycoside portion that is soluble in water, that makes them soluble in both categories of solvents. Okay, polar as well as non-polar, water as well as organic solvents. Then definitely they can give the chemical test like reducing Fehling solutions after the hydrolysis. After the hydrolysis, remember this like last few words. After the hydrolysis, definitely they give the chemical test for Fehling solutions. Then if you just talk about the stabilities of glycosides, the stability depends on the hydrolysis of acids action, hydrolysis of alkaline actions, hydrolysis of enzymatic actions. So on acid hydrolysis, if you add HCl or some organic acids, citric acids and lactic acid like that, the acid splits sugar and aglycone. Definitely each and every hydrolysis, acid, alkaline or enzymatic hydrolysis. Chemical hydrolysis, I can divide acid or alkaline into one portion. Chemical hydrolysis or enzymatic hydrolysis, that's going to separate the aglycone and glycone portion. Very obvious over here. But acid hydrolysis, it cannot separate the C glycosides as such. And from this point, from this line, from this slide, many times question has been asked into competitive examinations, which glycoside do not give the hydrolysis on acid hydrolysis methods. C glycoside, they are resistant to acid hydrolysis. Okay, then alkaline hydrolysis, where what happens if you just see the hydrolysis of esters group like lactanosides A to purpurea A, that is actually present in digitalis plant. Okay, and then there is also opening of various ring structures like lactone rings that can open under alkaline hydrolysis. And in our like physiological pH, if we see, we have both kind of acids as well as alkaline, both kind of pH are present at various organ systems or at various tissue levels. Then we have enzymatic hydrolysis. Definitely the enzyme will start separating the sugar step by step from terminal sugar, then it will come to disaccharide, and finally it will separate the monosaccharide. Okay, and all plants producing glycosides have enzymes that can hydrolyze these glycosides. All plants. Okay, you have studied about plant enzymes. So all plants have one category of enzymes which can actually hydrolyze these glycosides into them as such, whenever plant is under stress, whenever plant requires sugar, whenever plant require the aglycone portions, at that moment of time, the enzymes which are present in plant, they can separate it out. Okay, if I just tell you few more examples, like we have salicin. And salicin, if you just see the structure of salicin, it is very, very similar to today's salicylic acid. And then we again modified the salicylic acid into acetyl salicylic acid. Then it became very like very mega or very blockbuster molecule into the category of analgesics. Okay, now we have NSAIDs, categories of non-steroidal anti-inflammatory drugs, categories which actually derived from salicylic acid itself. So salicylic acid, let me tell you, that was initially pointed out in plants itself, and now you can see the structure of salicin that is present in salix bar, or that is a alcoholic and phenolic glycoside, right? Then we have arbutin and methyl arbutin, and the structures are also mentioned alongwise. And we have alkaloid glycoside, which we have just discussed, that is actually present in green vanilla pods, and you can see over here the structures of vanillin or glucovanillin, and you can also see the plants. Okay, then we have one category of glycosides or compounds where the CN cyanogenetic groups are present, or that liberate HCN gas on hydrolysis, and the higher concentration of HCN, that can be poisonous. Okay, you know, cyanide group CN minus, that can be poisonous if we have like sodium cyanide, potassium cyanide. So potassium cyanide is again like it's poisonous or deadly poisonous. Cyanide is itself like it's a topic of interest to study about that. In various societies, even like you already know Hitler, and they have all used cyanide poisoning for suicidal things. And similarly, the plants, they also have cyanogenetic glycosides like cyanophore glycosides, which are present in plant. On O-glycoside, they yield HCN gas on hydrolysis, amygdaline and linamarin. Okay, and now you can see the structure. From structure you can find out like these plants, what they will do, because of this CN group is present, they can release this on the hydrolysis as such. Okay, that is present in aglycone portion. Okay, this is the 1,6 linkage is there, and you can see over here this is a disaccharide sugar which is present, right? And now similarly you can also see like so after cyanogenetic glycosides, we move to next category of glycoside, that is actually the sulfur containing glycosides. And sulfur containing glycosides, they are present in various seeds also, and sulfur that is also present in mustard seeds as such. And thioglycosides or thioglycosides, glucosinolates, which are present in snigrine, which is present in various seeds as such. Because if you can see the association over here at this juncture is because of sulfur atom which is present between glucose and aglycone. Okay, then very common plant which is digitalis. Okay, digitalis lanata, digitalis purpurea, and this is one of the significant categories of herb or plant which we are using even today nowadays also for various heart related problems. Okay, decoctions, digitalis plants, we are using digitalis lanata, digitalis purpurea for various heart related problems. Okay, and similarly we have licorice as such, and in next class we will discuss about digitalis and licorice. and the basic introductions of glycosides, their definitions, their properties, their classifications, their examples of each and every category, that is over. Okay.